Hello, everyone. Welcome to my talk. First, I'd like to apologize that due to a time conflict, I cannot give this talk in real time. So I pre-record my presentation and ask the seminar moderator to play it for on my behalf. So I apologize for that. My talk is about surface enhancement Raman chemical imaging, reviews co-localization of pectin and xyloglucan inside onion epidermal cell wall. My name is Dr. Qianzu Yu from Department of Agriculture and Biosystem Engineering at Iowa State University in the United States. So first, I'd like to talk a little bit about the motivation behind the work that we did. As we know, plant cell wall is the first barrier to protect the plant. No matter it's bacteria or fungi, the pathogens that will invade plant will need to pass this barrier first. And typically, they secrete different types of enzyme to break through the plant cell wall by hydrolyze different polysaccharide in the cell wall structure. And to understand this, obviously, it's very important. Because if we understand, so what unfortunately as of today, right, we are not 100% understand the cell wall structure of plant cells. So by that we mean is we know what is in there. We know the different types of polysaccharide, cellulose, hemicellulose, pectin, that are in the plant cell wall. But what we don't know is how these different polysaccharides are layered or organized together to form the cell wall structure. When a pathogen invades the plant cells, it needs to go through the cell structure layer by layer, right? So if we understand this structure better, and from a breeding point of view, from a molecular engineering or genetic engineering point of view, that we can try to alter the cell wall structure. If we flip, for example, put a bottom layer to the top layer, then a regular pathogen, regularly when they invade plant cells, they need to break down the first layer first. But if we change that structure, then the enzyme that the pathogen will be secreting to break down the first layer won't be effective against the altered first layer. Then it will offer a good protection for the plant cells against the pathogen invasion. So that's the basic motivation behind this. If we understand the structure better, then we can pass the information down to biologists who are doing genetic engineering kind of breeding, that they can change the plant cell wall structure to offer better protection. And as I mentioned, until today, although we know what are in the plant cell wall, those are mainly polysaccharide type polymers, cellulose, hemicellulose, and pectin. But we don't really know how they are organized together. So that's one of the goals that we wanted to achieve in the study, is to develop a method that allow us to better understand this. In general, as you can see here, pectin actually and cellulose are the main components of plant cell wall. I mean, hemicellulose obviously is also a pretty significant component in there. So the method that we choose to develop is based on surface enhanced Raman spectroscopic imaging. This method has been used by others as well. Let me move this out of the way. See if I can move this out of the way. No, I can't. All right, so sorry. Let me go back. But in the past, what people have been doing is to use Raman to look at different species of polymers. For example, as you can see here, this is a figure. This is a figure from a published paper by Zheng Group. And they use the Raman imaging to look at xylem distribution in a plant cell wall. But what is not possible in the past is we cannot really use Raman imaging in the past to reveal co-localization of different types of polymers. how do they organize? Where are the places that this cross-linking or co-localization happen? That we don't really have a good method to reveal in the past. So what we developed, as you will see from the later part of this talk, is that we developed a technique that will allow us to look at the co-localization pattern. And for example, the results that I'm presenting today, we looked at the co-localization of pectin and the xyloglucan in onion cell wall. And so I'm gonna go into that. There are few technical difficulties when it comes to Raman spectroscopic imaging, and we come up with solutions to address each of these difficulties. First is what we call specificity issue. That is the main components in plant cell wall, these are polysaccharides. So they have similar bond structures. Although their Raman spectra are different from each other, but there are similarities that make it difficult to distinguish between different types of molecular species. For example, if you look at here, so this is pectin, this is xyloglucan, this is cellulose. These are the three main components in plant cell wall structures. And here is the spectrum collected from tomato skin. What you can see is, although we can identify peaks in the cell wall structure, but it's not that easy and simple to pinpoint peak to specific molecular species. And when you collect a cell wall structure, a cell wall spectrum like this, and also it's hard to really associate this information to spatial positioning, so that you can reveal that what polymer is at what spatial location. That is to say that it's hard to reveal the actual chemical structure of the plant cell wall. So what we developed to address this issue is we developed a in situ enzymatic hydrolysis. I will explain that later, to offer better specificity so that we can look at specific polymer species in the plant cell wall structure. And also there is a sensitivity issue because Raman scattering is a weak phenomenon. And in order to get better signal from the measurements so that we can use that to construct a better image, a chemical image of the plant cell walls. And we used a labeling technique associated with surface enhancement. And I'm going to go into that in more detail later as well. And also there's a resolution limit issue because the spatial resolution is limited by diffraction limit. And we developed nanofocusing, although I'm not going to talk about that in this talk. That's something that is under work right now. So the first thing is we developed a chemical labeling technique to label specific polysaccharide species, and then that is the pectin species in plant cells with a strong Raman scatter called 4-ATP. That's a 4-animosyl phenol. this guy here. This is a strong Raman scatter, and we can form a chemical bond between the amino group on the 4-ATP and the carboxylic group on the pectin backbone, so that this Raman tag can be attached to pectins, which offer much better spectrum signal intensity. As you can see here, the species that we're targeting mainly are the homogalactoronin, Hg. This is the main components in most of the pectins that you find in plant cells. And also Rg1 and Rg2 are the other two species that this tagging technique can work. And these pectins are abundant in plants. They make up to 35% of the plant cell wall material. And their base unit is the galacturonic acid. So they have the carboxylic group that is needed for attaching 4-ATP2. And we can use EDC and HS facilitated coupling reaction to attach 4-ATP to this pectin species, and which will improve the signal intensity. As you can see here, before and after 4-ATP tagging, we can clearly see here these two are the 4-ATP peaks that coming from the carbon-sulfur bond and the carbon-carbon bond of the 4-ATP molecules. And then with this very intensive peaks, it's easier for us to identify the location and presence of 4-ATP when they are conjugated onto the pectins, which will give us the location information for pectins as well. So that's the purpose of doing this tagging. And on top of the 4-ATP labeling or tagging, we are also using surface enhancement, in which a uniformly distributed silver nanoparticle layer is created on top of the plant cell wall structure that help us to further increase the specific peaks that we are observing from 4-ATP, so that give us a much better spectrum resolution and the signal intensity for us to generate images that will be effective for us to identify the co-localization of different species, polymer species. And as you can see here, before and after the nanoparticle enhancement, the intensity of the 4-ATP labeled ramen cell, the 4-ATP labeled onion cell wall is increased significantly. And it should be noted that the enhancement sometimes is ununiform. So we worked on that, and we developed a silver particle based enhancement that can generate more uniform enhancement across the board on the plant cell wall surface to address this hotspot non-uniformity issue. Because what we wanted to get is a chemical image of the entire cell wall structure, not just a piecemeal spot by spot. So we need to have a uniformly distributed enhancement across the board. And we were able to do that. So here is the idea of the in situ enzymatic hydrolysis. So suppose that this red lines are the pectin species are labeled with 4-ATP, right? And if we use an enzyme that specifically hydrolyzes those species, then after the in situ hydrolysis, these polymers are being chopped into small segments that become water soluble, so it can be washed away. So then if we measure again the spectrum, the signal coming from this pectin species, ATP labeled pectin species, will disappear. Okay, so if we then compare the before and after hydrolysis signal, that the differences will reveal where this pectin labeled, the ATP labeled pectin species are at. So this will give us the signal that we need to localize, to locate where the pectins are at. And from which we can generate a chemical image of the pectin distribution inside the plant cell wall. So that's the idea. And the specificity towards the pectin species is offered by the enzyme that we use. So if we use different enzymes that can hydrolyze different types of pectins, right? And the before-after signal will give you the localization information for those specific pectin species that we're trying to identify. And that is the specificity that we wanted to get. If we don't use the enzyme hydrolysis, just measure the spectrum, pectin species, pectins pretty much give you the same type of spectrum. So it's not possible to distinguish between, for example, the HGs with this RG1s with the RG2s, right? So by doing the hydrolysis by specific enzymes, we will get the specificity that we need to identify individual pectin species. And The enzymes that we choose to use in this study are two types. One is the EPG, that is the enzyme that chops down the 1,4-D galactosideronic linkage. This is a backbone linkage in the polygalacturonians, the PG, right? So once we chop that down into smaller pieces, they're being washed away, that we compare the signal before and after hydrolysis. We can identify the presence and the location of the PGs in the plant cell wall. So that's what we're doing here. And later on, I will show the results that we obtained by using a second enzyme to go along with it. So I will explain that when we get there. So here is the experimental setup. First, we labeled our plant cells, the onion skin cells. That's the one that we choose to study in the study with 4-ATP. And then we use the silver nanoparticle-based enhancement technique. We wanted to compare the difference that it makes before and after the nano enhancement to see whether the surface enhancement is necessary for us to carry out this job out. So here shows the difference before and after the nano enhancer is applied. And so the first, here, okay. Yeah, so the first panel here is the fresh onion. So basically this means that the onion is not being treated with the nano enhancer. And then here, and 4-ATP. And then here is the 4-ATP labeled nano enhanced onion skin cell surface. Right, so what we can tell is the differences is clearly identifiable coming from the two ATP peaks before and after hydrolysis. Remember, after hydrolysis, the PG, the backbone of the pectin that are 4-ATP labeled are being chopped into smaller pieces, and the segments are being washed away. So the difference between the red and the green tells you where the specific enzymatic reaction have occurred. And those are the positions that we can pinpoint to presence of the pectin at those locations. And because the 4-ATP labeling and the nano enhancement give us a much significant difference that can be observed, right? Change before and after hydrolysis. So this will offer a better chance of revealing where the 4-ATP labeled pectins are at. And we also looked at whether this so-called in situ hydrolysis reaction are specific towards pectin. So the first panel here is a comparison between peaks that are associated with cellulose. Before and after the ATP attachment with the nano enhancement, you can see there is no significant changes for the peaks for the cellulose, which means the enzyme is not really hydrolyzing cellulose, which is what we expect. And here shows the difference for the 4-ATP peaks. Again, this is 4-ATP labeled pectin. This actually is a specific pectin peak that we're using here. And what we can say is, oh, yeah, let me go back. So this is a specific pectin peak that we are comparing between the treated onion cell wall and the fresh onion cell wall. Since this is a pectin peak, we do expect a similar trend be observed, right? There should be changes because those changes are caused by the hydrolysis, the in situ hydrolysis. And the result shows that that's what we observe. Right? There are significant changes here. And the third is for the 4-ATP peak. Again, 4-ATP labeled with nano enhancement, we can clearly see a very significant change in the 4-ATP peak. There's no such change observed in fresh onion because there's no 4-ATP for the fresh onion, right? And the reason why we're comparing this is you can see the scale of the intensity change for 4-ATP is almost double the scale of the pectin peak change. So higher change means we have a better chance of identifying those changes in the images to create the image that we need to analyze the localization pattern. And so those are the background of what we're doing and what the methodology is. And the next, I'm gonna show some of the results that we have obtained. So in a nutshell, we wanted to find out about the colocalization of pectin and hemicellulose. And we wanted to be able to obtain a higher sensitivity and a higher specificity as well. So sensitivity improvement is obtained through the surface enhancement, and the specificity improvement is obtained through the in situ hydrolysis. Right, so here again shows that we, like I mentioned early on, we developed a silver nanoparticle-based uniform enhancement technique to obtain uniform enhancement throughout the sample surface we're analyzing. And now, here shows some of the results that we have obtained, right? So here, the first panel here, these are optical microscopic image of a onion skin cell. You can see the cell walls here. And then with our technique that we developed, that we looked at different locations throughout the cell wall structure at the corner versus in the middle, and to identify where that we're observing the most change before and after hydrolysis. Again, remember those changes associated with the distribution of specific pectin species that are being labeled and that are being hydrolyzed. So clearly here shows that at the corner regions, especially the cell wall that is connected to the middle lamella region, that we observe the most change in signal, that is the indication of highest presence of the pectin species in question. And then we also, as I mentioned, use the second enzyme, which is the XEG, and this enzyme will hydrolyze away xyloglucan, which is a hemicellulose. So the purpose of this is, in certain locations, we know that the hemicellulose, like a xyloglucan, is bundled together with pectins. So in those regions, the pectins initially are protected by those hemicellulose against hydrolysis. And if we hydrolyze away the hemicellulose first, which will expose the pectins for further enzymatic hydrolysis. And if we do this in sequence, that will allow us, by analyzing the changing signal before and after hydrolysis, to identify those regions where colocalization of hemicellulose and pectin are occurring. So those are the regions that we will see, okay, these are where the cross-link or the crossover between hemicellulose and pectin are happening. Okay, and that will give us the chemical information that we need eventually to reveal the organization of different types of polysaccharides in the cell wall. So that's what we did. First, we looked at the EPG hydrolysis pattern as I said before and after hydrolysis. It will reveal the distribution of pectin in the cell wall structure. And clearly that we're seeing that here, that before and after hydrolysis, you can see that the region where the pectins are being hydrolyzed away. And again, you can see the pretty strong change happening in the corner region. Right? And that's consistent with what we observed in the previous slide. And then what we did is we hydrolyzed the same type of cell wall structure with XEG first. Okay, that will chop away the xylaglucan, the hemicellulose, and expose those otherwise protected pectin locations for further hydrolysis, for the EPG hydrolysis to happen. Right. So here is what we did. We first exposed the surface with the with XEG for 30 minutes and look at change in the Raman intensity based on the Raman intensity change. Again, this Raman intensity are coming from the 4-ATP label that we conjugate to the pectin and hemicellulose molecular species, so that by looking at change in 4-ATP intensity, then we can get information regarding the distribution of this labeled molecular species in the cell structure. Then we treated the sample after XEG treatment with EPG again. So further intensity decline means that where this intensity decline happens is the region where the pectin used to be protected by the xylaglucan, by the hemicellulose. Now, since the hemicellulose are being removed, the pectin is exposed, so that the hydrolysis will kick in and chop those pectin chains down. And when the segments are being washed away, then significant changes in intensity is observed, from which we can deduce here, and we can identify those locations where the co-localization between hemicellulose and pectins are happening. And by analyzing the data collected by generating this chemical image, these are chemical images generated based on the intensity change, and we can clearly identify regions that the highest decrease happens at the outlayer of the cell wall that connected to the middle laminar area. So, which tells us that those are the regions where the xylaglucan and the pectin are co-localized. And co-localized in this context usually suggests there's some cross-link between xylaglucan and pectin species in those regions. And those regions are also where the two species are protecting each other. And so if, for example, a specific pathogen uses enzymes to break down the cell wall structure, those might be the areas where the protection is strongest against the enzymatic breaking down. And which means that if some kind of genetic engineering technique can be applied to alter where these locations are or should be at to offer better protection for the plant, and this information will be useful for monitoring before and after whatever genetic engineering technique is being used, whether those alteration is effective or not. Right, so again, what we gather from our observation is for onion skin cell, the cell wall structure has the highest pectin in the corner in the middle laminar area. And in the region where the coexistence of xylaglucan or hemicellulose and pectin are observed, also in the cell corner in the middle laminar area. And so this reveals that where the co-localization are at for these two important polysaccharide species in the plant cell. So as a summary, what we did was to develop a technology that will allow us to review colocalization between different polysaccharide species in a plant cells, plant cell structure, which is otherwise not achievable with the regular spectroscopic imaging or other imaging technique. Then our results indicate there's no significant pectin and zelloglucan interaction at the cell surface and at the corner, but those colocalization mostly occurred in the areas that connects to the middle lamina area and in the middle lamina area. So those are the areas where the colocalization between pectin and hemicellulose, the specific hemicellulose, the zelloglucan that we studied in the research, that is happening. And so in general, this technique is useful for us to understand plant cell wall polysaccharide organization pattern and help guide further research into understand the overall structure of plant cell wall. Okay. So at the end, I'd like to acknowledge my collaborators, Dr. Zabotina at Iowa State and Dr. Xinwei Wang at Iowa State, and also the grad students that are involved in this research, Qing He, Hongxu Wang, and Bingbing Zhu. And we also wanted to acknowledge the sponsorship from USDA NIFA, grant number 2015-07802 for support this research. Thank you for listening to my presentation. And I apologize again that due to a time conflict, I cannot be there to answer questions. But if you have questions, please reach out to me. And you can email me, but I'll be more than happy to answer them. Thank you.